to give you idea about the different aspects of bioinformatics. Specifically, you will get to know how to build the structure of a molecule. So, before moving to that, let us have a brief recap of what can be done in bioinformatics. So, this is bioinformatics. I think all of you know what bioinformatics is because this portion is in your syllabus, I believe. So, it is kind of an amalgamation of different biological fields and the solution to the problems of biological fields using computers. So this is basically the NSF mission statement. So it says that how to expand your knowledge, how to expand your biological knowledge, or how to do problem solving with the help of computers. And the problems are specifically biological problems. In bioinformatics, we take a top-down approach. Like we can have the structure, we can extract the sequence from the structure, we can have the sequence that can be converted to the structure. So both ways we can do, both the things we can perform. And so basically, in bioinformatics field, we are interested in the sequence and structural information. For sequence, we have the nucleotide sequences, and for that means for proteins, we have the amino acid sequences. And regarding the structures, we can build the structure of a protein from its amino acid sequence. And the method is called comparative modeling or homology modeling that we will learn today how to do that. So these are some basic things, the different amino acids, because while building the models, you should have a very good idea about the different types of amino acids that we have, because the nature of the amino acid guides the structure. So, we all know the structure of a protein is so important. Why it is so important? This is important because the structure defines the function. The structure defines the function. So, if we can predict the structure of a protein, we can somehow predict the function. So, here a word of caution is there. We always predict. We cannot say it is 100% accurate, but the methods so far are fairly accurate to actually reproduce the experimental data. This is us very basic biology, so what proteins do, and that means the proteins are enzymes. There are structural proteins like collagens. There are transport proteins. And these are the different levels of protein structures. The primary structure, that means the sequence of amino acid residues. The secondary structures, it is the local folding of the primary structure. The tertiary structure is actually the three-dimensional structure, and quaternary structure. This is valid for the proteins having multiple chains. That means relative orientation of one chain with respect to the other. That is called the quaternary structure. And this is not that important. So now let us move on to how the sequence looks in a computer. This is a nucleotide sequence that you will get to know today. This nucleotide sequence, this is the single letter code of the nucleotides. And the first line, it is called the FASTA format. It starts with this protein sign. And first line is kind of the description, because in the first line there is this Homo sapiens hemoglobin. This is the nucleotide sequence of the alpha chain of hemoglobin from human urine. And that one, that means the number 144 and then NN, all these stuff, all these are called the accession numbers. That means these are kind of the pointers towards this particular sequence. And this is the amino acid sequence which is coming from this. So this portion, the red portion, represents the actual gene which is encoded into the amino acid of the protein. And this is the entire genome. This an entire genome looks like this. So, yes, we will come to this portion. But before that, let us have a demonstration of the different databases. So now Dr. Shima Vishwas and Torusri Mukherjee, they will demonstrate the different databases to you. So listen to them carefully and how to access the databases and what information you can gather from the databases and how to download the sequences from the databases. They will show you all those stuff.
So databases are those kind of those platform where we can store the data, like any kind of data, sequence data, three-dimensional data, and some metabolic data, some small molecule data. Those are databases. Basically, databases are divided into two broad groups. The first one is the primary database, and second one is the derived or secondary database. What are the primary database? The primary database are those database where the data are directly submitted by the scientist, and those data are derived from the experiments. Suppose you do some experiment and you have done some sequencing and generate a sequence, and you can directly, if you directly submitted that sequence in a database, that is the primary database. Like what are the example of primary database? DDBJ, where we can store the nucleotide sequence, and those databases are derived from the primary database. The data are derived from the primary database, but some modification have been done because lots of data are available, but some data are not required for the for further experiment, or there are some missing information. Those information are filled and deposited to the secondary database or derived database. So here we will see the UniProt database. It is basically a secondary database where the amino acid sequence are stored. If you search into the Google and type UniProt, you can do it in your phone. If you want to do it, you can do it in your phone. So first, go to the Google and type UniProt, and then press enter. Here you can type anything. What is your query? If you type a protein name, this page will be open. In the left side, you can see the status. This data derived from Trimble and Swisspro database, and the organism name is present here. Like hemoglobin, in human hemoglobin, 210 data are available. In mouse hemoglobin, 108 data are available, and so on. You can click in human, then the human hemoglobin data will be displayed. If we click in this entry, then this page will be open. So in UniProt, various type of information are available, like what is the function of the protein, the name and taxonomy of the protein, the subcellular location of the protein, and if this protein is associated with some kind of disease and some variant, then this information is also available in the UniProt database. Then the expression data are available, and then the interaction of this protein with other proteins information are also available. And most importantly, the structure of the protein where we do with the hemoglobin. So hemoglobin structure is also present here. And the family and domains, you can check what type, how many domains are present in this protein, and then the sequence of the amino acid sequence of this protein is available also in the UniProt database. So this is the accession number of the human hemoglobin protein. This accession number is also the identifier of the human hemoglobin beta subunit, and this accession number is very important because it is the identification of the human hemoglobin beta subunit, and in every entry there will be different accession number. So in this section we can see that this the protein name is hemoglobin, human hemoglobin, and the subunit is beta. And also here the gene name is present. If we go to the function section, then we can see the function of the hemoglobin is also written here. The binding site information of hemoglobin protein is present here. In the sequence section, we can see that the human hemoglobin beta subunit length is 147 amino acid, and the amino acid sequence is present here. We can download the amino acid sequence from this UniProt database. Just click the download option, and then the sequence will be displayed. Copy this sequence and you can save it. You can save this sequence file as FASTA format. Just paste the sequence in a notepad and save this file as hemoglobin beta subunit .FASTA. The extension of any kind of sequence file is FASTA. So So in the structure section, we can see that there are many three-dimensional structure are available for the human hemoglobin beta subunit. We can see the three-dimensional structure of the hemoglobin protein. This is the PDB accession number of the human hemoglobin, and this section and the information, all information about the human hemoglobin beta subunit is present here. This is a mutant variant, and here the DOI is also present. From this DOI you can access the paper and the experiment by which this three-dimensional structure is tried. And from here you can see that which method is followed to detect this structure is also demonstrated. We can get the three-dimensional structure of any protein by the help of X-ray crystallography, NMR, and by these techniques. And the resolution of this protein is 2 angstrom. And the experimental details of this protein is also present here. And the literature information of the human hemoglobin is present in this section. The chain information, the sequence length, and the from which organism it is derived, this information are available in this section. If we go to the structure section, we can see the sequence of the human hemoglobin beta subunit. The administration section is very much important. In the scope database, we can check the class of this protein or any kind of protein, the fold of the hemoglobin protein, like the hemoglobin is very like protein, and the family information and domain information of any protein. CAT is also the similar type of database where we can classify the protein, the class of this protein, and the architecture, topology, and homology of the protein is also information is also present here, and we can retrieve this information from the CAT database. The experimental details of this protein is present here. The protein is obtained from the X-ray diffraction technique. The sequence information of the hemoglobin protein is present here, and this by the help of this picture we can say that this portion will generate alpha helix, and the three-dimensional view of this portion is also alpha helix. And the genome information of hemoglobin protein is also present in the protein data bank. If any kind of ligand is interact with the hemoglobin protein, those information are also present in the protein data bank. view of the of this protein by just treating the PDB form. PDB file should contain the sequence information, the chain information, and their coordinate information.
Okay, let us have your views. So, so far, is there any problem, or you want us to clarify again regarding, because these are the basic things that we need to know first. Then only we will be able to move on to the modeling techniques. So, you understood so far what Dr. Vishesh was talking about? Okay. So far, what we learned, till now, what we learned about the database, and we got to know what type of information are there. Now, can you download a data from there? Will you be able to download a data from there? All of you? You can download the data from there. Now, the next question is, will you be able to understand what the data are? The different annotations that are there, the different information that are there. You can understand? So, in your exam, like in any practical examination, if you are given an accession number, so will you be able to analyze that accession number? You can do it, all of it. Very good. So, if you have any question or any doubt, you can tell me. Anything that you want me to clarify? You understood the FASTA format? You understood the different sequences? You understood the annotations? Annotations are very important. Annotations means, see, if you have only the sequence information, that will take you nowhere. You won't be able to do anything until and unless you know what information that sequence is giving you. For example, in UniProt, there are annotations like post-translational modifications. To which particular amino acid the post-translational modification will occur. Right? So, these are the annotations. I think they have not yet received the feedback forms. So, anything that you have in your mind? Okay, fine, very good. Now, we have the data. We have the data with us. We are mainly focusing on the amino acid sequence. We are not considering the nucleic acid sequence that much here. Why we are not considering the nucleic acid sequence? Because nucleic acids, they code for the amino acids, but the amino acids are converted to, or the amino acids build up the protein, and proteins are the functional molecules. So, we are interested in the features, protein features of proteins. We are interested in that. And so, we will learn the technique that how to develop or how to build the structure of a protein from its amino acid sequence. That method, these are the methods for that. The first method is called homology or comparative modeling. The second one is fold recognition. Third one is ab initio method or knowledge based. And the fourth one is ab initio method without knowledge based. These two are very difficult. And the first one is the most accurate one. Now the question is, why? The first question is why? Why should we go for this type of structure prediction methods? Why? That is because there are different techniques. I think all of you know the X-ray crystallography. Have you heard of it? NMR. There is cryo-electron microscopy. Okay. All these are the methods to build or to generate the structure of a protein or a biomolecule rather. Right? Since all these methods are available, why should we go for bioinformatics technique like this? The thing is that number one is it is not possible to have the structure of a protein by all these methods, because for X-ray crystallography, the protein, you have to make a protein crystal, which is not always possible. The second one is for NMR. NMR is basically valid for proteins having amino acids 100. That means more than 100 amino acid residues. If a protein has more than 100 amino acid residues, generally the NMR is not a good method. And the other methods, all the other experimental methods, the wet lab based methods, they have their own shortcomings, own problems. Now, if we cannot crystallize a protein, if we cannot solubilize a protein, how will it be able to perform the crystallography or NMR? We won't be able to do that. In that case, we have to perform bioinformatics analysis to build their structure, right? Now, the basic principle of generating the structure of a protein from its amino acid sequence is the homology modeling, and its principle is protein structure is more conserved than sequence. Protein structure is more conserved than sequence, number one. And number two is the sequence determines the structure. That means if two proteins have certain levels of sequence identity, certain levels of identical amino acid residues, the two proteins are considered to have similar structures. The cutoff value is nearly 30% or more. So if two proteins have amino acid sequences which are 30% or more identity, in that case, those two proteins are considered to have similar structures. Right? The more the sequence identity, the better is the generated structure. Right? So that is the basic thing for homology modeling or comparative modeling. Yes. And these are the things that I just discussed. These are the things. So the three-dimensional structure is important. The three-dimensional structure is important to identify the overall functionality of the protein. So if you can have the amino acid sequence of a protein, what can you do with the protein? You can predict its structure with the help of bioinformatics tools. And from the predicted structure, you can identify its possible function. So these are the basic steps of homology modeling. First is template recognition. Now, what is a template? What is a template? Template means it is a protein. It is a protein which has 30% or more, or at least 30% sequence identity with the protein for which we are going to build the structure. That is called template recognition. Template. The next one is alignment. We have to identify the percentage of amino acid residues that are identical. That is called alignment. Then determine the structurally conserved regions. In case of proteins having similar sequences, there are certain regions which are structurally conserved. For example, if we can consider the DNA binding proteins, you remember DNA binding proteins, they have a helix turn helix, or a leucine zipper, or a zinc finger motif. These are called the structurally conserved regions. And those structurally conserved regions have identical or nearly identical amino acid sequences. So, between the target means the unknown protein, and the template means the known protein. Between the target and the template, if we can identify those regions which have a similar type of amino acid disposition, similar type of constant stretch of amino acids, we can consider those regions to be structurally conserved. Okay, everybody got my point? Then backbone generation. This is why I told you that you need to remember the structures of amino acids very carefully. Why? That is because the backbone, for all the proteins, the backbone is nearly the same. There is this carbon atom, one carbon atom, double bonded oxygen. If I can write it here, that is continuing to do. Everybody can see? This is the C alpha carbon. This is the carbon, carbonyl carbon. This is the nitrogen. This is again hydrogen, and there are R groups. And so on, this process continues. So this particular structure this particular arrangement is nearly constant for all the types of amino acids and for all the types of proteins. What varies is the R, the presence of the R. That varies. So we can simply make the backbone because it remains constant for all the proteins. But what varies are the R groups, the side chains. Then comes loops or variable regions. This is a little bit tricky thing. See, if we consider a loop, there is this alpha helix, and this is the loop region. This is the loop region. Now, the loop regions are flexible. It can have any structure. So it is really difficult to generate the loop. Now, why this is so? Why we should be very careful about building the structure of a loop? That generally loop regions remain exposed to the cellular environment, and the cellular environment is mostly polar, water. There are water molecules. So the amino acid residues that are present in the loop region, they should be the polar amino acid residues because they should be facing the polar environment, right? But now, if in your target sequence, if you have non-polar amino acid will you be able to fit them in the loop regions, or will that be biologically meaningful? No. Biologically not meaningful. But what happens? What happens if you find those amino acid residues in a loop? Now you have to use the method of artificial intelligence, AI based methods. So what AI does is that, okay, there may be a value, there may be an isoleucine here, but their side chains should be, not side chains, sorry, their carbonic groups or the polar portions should be exposed to the surrounding polar medium, and their side chains should be interior and packed together in such a way that they kind of exclude the water. Right? There comes the bioinformatics and the artificial intelligence. Everybody got my point? Yes. Because we all heard of the term artificial intelligence, but we don't know what artificial intelligence actually does. Artificial intelligence is useful in making such decisions, right? Then I'll change. What is actually meant by the conformation and such? What is actually conformation? Conformation, there are two terms, conformation and configuration. So what they do is they are momentary arrangements of atoms or groups of atoms, but conformation, one conformation can be converted into other conformation. That means they are movable, but configuration is fixed. Now, conformational search of amine, that means the side chains. So it may so happen that, say, there is another R1 and R2. Now, this R1 and R2, these two side chains should not be so close that they have some kind of unfavorable interactions. That is called steric hindrance, right? That is called a conformational search for the side chain. The side chains should be placed in such a way in order to avoid the unfavorable interactions. Right? Then, structural refinement. Structural refinement means, okay, we build the structure. We calculate the energy of the structure. Right? So, how many of you know what Gibbs free energy is? Gibbs free energy, all of you heard of it? Yes. Internal energy? Yes. All of you heard of it? So, we calculate first the internal energy. Right? So, from our thermodynamics point of view, we all know that the change in internal energy, delta U, right? That has to be negative for a structure or for a molecule to be stable. Right? So, refinement of the structure is that we have to have a structure for which the energy is the minimum, the energy is the most negative. That is called the refinement of the structure. And validating. Validating the structure means we build the structure. We have the structure. Now we have to compare the structure that we built with the already known structures that are available in the database PDB. We have to compare the two. So that we can identify how accurate the structure that we generated using the bioinformatics technique is. Okay, so it is called the validation, structure validation. So these are the methods of homology modeling or comparative modeling. So see, in the first place, I told you that bioinformatics is an interdisciplinary subject. It involves knowledge of all the subjects. So Here we have to have the knowledge of chemistry, we have to have the knowledge of physics, we have to have the knowledge of biology, as well as we have to have the knowledge of artificial intelligence, that means computer science. So that is why it is an interdisciplinary subject, but the usefulness lies here. Using the principles of all these subjects, we can solve a biological problem like this. So this is the method of building the structure of a protein from its amino acid sequence. This is a very, very important method. Why this is a very, very important method? Because these structure prediction methods are used in a variety of experiments as well as the drug designing, for drug designing purposes. Okay. So this is comparative modeling or homology modeling. Now, Tanushree Mukherjee will describe the process to you.
As I have said that the processes of modeling, homology modeling is the most famous method where we use a template and we gather the information from data repository named as PDB. So as for homology modeling, we have to acquire the sequence of the target sequence of the protein, which protein we are working on. Okay. So for that, we have to obtain the sequence from Uniprot, and then we have to place it in a server named as Swissmod. So this is the sequence of hemoglobin, which has been obtained from Uniprot. So we will copy the full sequence. Remember, the faster sequence of a protein is always denoted with these characters. These are important as well. So, this small.
All of you take it down, because you can use it with your phone or your laptop. If you have a good network, you can simply use this particular tool. So what I would suggest is, if you have your phone and if you have your network connectivity in your phone, so you keep on doing what she's doing.
This is the URL of Swiss model. It is a free server, and it helps us to identify and model certain sequences which have, as I have said, homology greater than 30%. Why? Because it is needed to understand and needed to find out from which sequence or from which species it has the homology. Homology means the similarity. So, after opening Swiss model, we will start doing modeling. So, we will click on this button, and this server provides a box where we have to paste our target sequence. Either it can be a full sequence of a protein or it can be a part of a protein sequence. So it depends on the choice of what we are doing. Okay. Suppose a protein that is 1000 amino acids long, we would like to know its structural patterns. So it is important to put all the sequences together here. But when we are interested in more domain specific work, like proteins are always not engaged in only one particular type of function. Multiple proteins, they can contain multiple domains, and these domains are important. They can be chopped off during ubiquitination or other cellular processes. So these type of domains are complicated as well, and they contain some structural informations other than the other domains of the same protein. So when we are working, it is our duty to know what kind of things we are looking for. So here I am going to paste the sequence, as I have said, with all the characters mentioned here from Uniprot we have downloaded. I will be putting all the characters here, and I will first search for templates. There are two other options are there. I will be going for search for templates first. The server is going through its data and other data repositories to find out what kind of sequence it is, and it will provide other homologous sequences with detailed information. It will take a little time. Just like Uniprot, it is also a secondary database. Like from this database, you can search for other options as well, and you can process the sequences. You can find out the other templates, which can be useful in other studies as well. Time. So this page contains some useful informations. See, these are the results we have got. The template result is that it provided templates each for each individual. We can see the PDB ID, the name of the protein, and the gene name, and the sequence coverage, that sequence identity. কত percent sequence identity আছে has been also mentioned. এখানে we have got so many options. Okay, so the first option, which shows the best match, we will go for that. Also the predicted structure, that is the best template for this chain, has also been shown here. Here, if you can hover the mouse here, you can see the secondary structure of this protein molecule, that is mostly composed of alpha helices. Okay? So we would like to open this first result here. After opening the first result we have obtained, see, there are so many informations. The sequence, the FASTA sequence is given, how many amino acids it contains, it is given, the identical species also. So it is very much important in which what kind of species we are looking for. Like when we are working for mammals or Homo sapiens, we would look for much Homo sapiens or mice model or rat. But here, see, this is Macaca speciosa, that is different from the thing we are looking for. So I would be doing build models first. So this was the template search. From this you have to identify what kind of species you are looking for, and then it will be providing all the results containing all the information. So if I go for build models, it will generate the model as required. Okay, so it is excluding these template searches and all informations. It is now preparing the model based on our query.
See, the template search that was shown in the previous page, that template search is the first step that I was talking about while I was describing the process of homology. Now, please go back to the... Yes, see, this is the sequence alignment. That means after you have given input of your unknown sequence. Here it is not the unknown sequence, it is a known sequence, hemoglobin, but we are considering that to be the unknown sequence for the sake of simplicity. So we use that sequence, considering that to be an unknown sequence. We use that sequence in the query box and perform the template search. So that search generated all these results. From all these results, if you just please go on. Yeah. If you see, these are the hemoglobin, structures of hemoglobin, right? But what important information that you need to remember is that your unknown sequence is from Homo sapiens. So the template has to be from a higher eukaryote, not from some lower ones. That information you need to remember, and while selecting the particular template, you need to remember that. Right? You cannot select any of the template. You cannot select just any of the template. You have to have that biological information. Otherwise, you will get a wrong model. The protein structure that you build using the bioinformatics technique will be a wrong one, will not be a correct one. Right? So these information you need to be very, very careful about. Another important information that if you scroll down, see, there are this X-ray 2 angstrom, X-ray 1.9 angstrom, and then again X-ray 2.9 angstrom, and so many structures are there with different values of the X-ray wavelength. You need to choose the template having the minimum wavelength. So between all these 1.9, 2.9 and 2, the 1.9 should be the best template. That is another aspect that you need to remember. But see, this one that is 1.9 angstrom, hemoglobin delta chain. But what was your input? It was the alpha chain of hemoglobin, or the beta chain, not the delta chain. So you can discard this one also, even if it has a good resolution, even if it is coming from human, but you have to discard it. Why? Because it is the structure of the delta chain and not the alpha chain. You are going to build the structure of the alpha chain. Right? So see, the process looks very simple, but it is not as simple. You have to be very careful. You have to keep in mind all these while building the structure. Right? So what three most important information that you have to consider? First is the species information. The second is the resolution. And the third is the type of sequence, type of input sequence. Right? Everybody got my point? Or anything you want me to clarify? Everything is clear? Right? So we can choose the template on the basis of all these criteria that I just mentioned, and then build or generate the model.
Sir, so what does that number signify?
Which number?
The mouse pointer and
Which one?
It is beside the blue.
0.9.
beside the blue word.
0.93.
0.93, that one.
0.93.
Yeah.
0.93. Okay. See. It is basically, this is a good question. It is basically gives you an idea about the percentage of sequence identity. So all these are nearly 93% identical. 93% identical. Okay.
But sir,
So the first one is 90.
but sir, when we scrolled, I mean,
The details.
when I go to the details,
Just go to the details.
Which one? Delta chain.
Anyone.
anyone. Details means that add over
The
that
drop
is
down.
pointing down.
Drop down.
The drop down.
This one, you want to open this one?
Yes.
Okay, this is also fine.
Yes. There the sequence similarity is not 97%.
See, there are two things. One is similarity, one is identity. Right? Two things are there. Similarity and identity. Similarity means, and this is, we are considering the entire stretch, entire sequence. And identity means we are considering some specific portions, like the active site region. For in case of hemoglobin, we are mainly considering the particular portion which binds to the porphyrin ring.
Then what should be my template in that case?
The thing is that yes, that's a very tricky situation. You have isolated a sequence, that means a protein, and you sequence the protein and you got the sequence. But you don't know, you can't find out the template directly. There are techniques to identify the template, the template recognition. But those techniques, it is not possible to teach you those techniques in just one workshop. So maybe in the next workshop we will teach you how to do that. That is a specific process that is called sequence alignment. So but that sequence alignment process is also a very complicated one. You have to consider many things while considering that's a sequence alignment. Here, this is a technique. This performs sequence alignment, but this sequence alignment that you are performing here, it is automated. That means you cannot put your input here. But because we are using hemoglobin, which is quite well known. But if you have an entirely unknown protein, we have to perform sequence alignment separately to identify the template. And that requires your knowledge of biology. Okay. Anything? That's a good question, definitely. Anything that you have in your mind? At least the idea is that you have an overview of the process so that you can use these techniques to build some small proteins, to develop the structures of some small proteins. That is the main aim of this workshop. So anything that you want me to clarify here? Now, everybody is getting my point and everybody can do this stuff, right? Okay, then go to
We will start doing modeling. Here, the best model that Swiss model has provided based upon the percent query coverage and sequence identity is this one. Here, it has also mentioned the template name, that is the hemoglobin subunit beta obtained from AlphaFold. This is also a data repository just like PDB, and the informations are there, and the predicted structure of the model is also present here, which we can download or which we can analyze further using other tools. So if I go down, the method is that Angshuman, as I mentioned previously, sequence similarity is 0.60, coverage is 1, and the range, that is the amino acid range, is 1 to 147. That is, the whole amino acid sequence has been covered using this method. Okay. Now, if we want to analyze further the structure, we can download this model. Okay, so structural analysis and assessment is very much important, because if we want to work further, the torsional angles that has been produced during this modeling has to be in a perfect orientation. Okay, from this the concept of Ramachandran plot has been established. Okay, the phi and chi angles that are important to address in case of protein structure, because this sequence structure leads to the functionality of a protein. If the structure is destabilized or it contains some mismatched phi-chi angles, it will not be fruitful in the cellular environment. So we will further proceed by downloading this model. Now this model is, if we go down from this drop down, we will obtain that the PDB format and other formats are there. So the best method is to download the PDB format, because it is the user friendly method. We can use it and we can open it into other molecular viewer tools are there, Rasmol, Bimol and other things. So after downloading, we will first check how this model looks like in a WordPad. It is looking, it has provided this alpha helical structure and all, but when we are opening into a WordPad, what will happen? See. So,
when you just mentioned earlier that the Ramachandran plot is something applied in this portion, and Saskia, which portion you have just mentioned?
yeah, I will go to that part. Here I am explaining the PDB structure, how it looks like in a WordPad. This is the structure of this model that we have got. There are the information of the atomic coordinates of the structure. These are the amino acids 1, 2, 3, 4 and so on. From 147 we can see the amino acid coordinates in XYZ. And this is the tar at the end. So this WordPad information combines into a pdb.pdb format, which is useful for identifying the structure. Now we will go for the structural assessment of this model. For that we will use ProCheck.
Okay, so before moving into that, let me explain one thing. We have built the model. We have generated the model with the help of the tool Swiss model. We downloaded the structure. Torushi just showed you the structure, opened it in a WordPad and showed you how the model or how the file looks like. Now what we are going to do is to verify or to refine the structure. So we will check whether the model structure has any amino acid residue in the disallowed region of the Ramachandran plot. So I think all of you are familiar with Ramachandran plot. So Ramachandran diagram or the Ramachandran plot is the plot of phi and psi angles, dihedral angles, and they give you the idea about the stability of a protein structure. If a protein has all its amino acid residues in the allowed regions of the Ramachandran plot, the structure is correct. If it has one or two amino acid residues in the disallowed region, then the structure is, you need to refine the structure. Because the amino acid residues, for a stable protein, the amino acid residues should be present in the allowed regions and not in the disallowed region. Okay. Now, Tonushri is going to show you how to analyze the protein structure with the help of this tool. The name of the tool is called ProCheck, which helps you to draw the Ramachandran plot from the structure of a protein. Okay, that is called ProCheck.
Sir, shall I use saves for that?
Yes,
It's a
saves
bit
also.
complicated.
Saves. Just explain what saves is.
Now, we will go further for the structural assessment of the built model. Saves is the server that provides the the quality and the physical attributes, molecular coordinates, and the Ramachandran plot that has been Ramachandran plot that should be assessed further, and Project provides the results in a user-friendly manner. So for that we will be choosing the file from the download model. Downloads. It should be a PDB file, that is .pdb extension should be there. After that we will run the program. Now, as I have said, there are other attributes and parameters are there to check the structure, but for the sake of simplicity, we will further proceed with ProCheck. Okay? Now, the question that you have asked, proteins are made up of peptide bonds, that is CONH, CONH, CONH. The bond is in a plane, all the bonds are in a single plane, and the C alpha and the other carbon atoms, they are facing just upwards and downwards, upwards and downwards. So, if we consider it is as an angle, it should be like in a particular fashion, so that it should be neglecting the spherical hindrance that is producing. So for that, the peptide bonds are producing a particular angle, phi and psi. এই দুটো পার্টিকুলার অ্যাঙ্গেলের ওপরেই এদেরকে চারটে কোঅর্ডিনেটে ভাগ করা হয়েছে রামাচন্দ্রন প্লট যদি আমরা দেখি সেখানে কিছু অ্যালাউড রিজিয়নস আছে যেটার জন্য শুধুমাত্র আলফা হেলিসেস যদি হয় বিটা শিটস হয় লুপস হয় বেন্ডস হয় তার মধ্যে থাকার জন্য যে পারমিসিবল রেঞ্জটা রয়েছে ফাই এন্ড সাই সেটাই হতে হবে আদারওয়াইজ দিস প্রোটিন ইজ নট অ্যাট অল থার্মোডাইনামিক্যালি আনফেভারেবল ঠিক আছে
ওখানে তাহলে আমাকে ওখানে চেক করতে হবে যে আসলে আমার যে অ্যাপ্লিকেশনটা মানে রামচন্দ্র পটের বাইরে কোনো কোশ্চেন ডিজাইনার রিজিয়নে
as I have proceeded through ProCheck only, it got me some result that I will be explaining further. So this is the result we obtained from ProCheck. We will further see the Ramachandran plot that will be useful for us. See, these things I have been talking about. There are 4 coordinates are there, and they are permissible allowed regions are there for the phi and psi angle. The best permissible, that is the most favorable region, is marked as red. Then the yellow one, that is the generously allowed or favorable region, and the white region is the disallowed region. If any amino acid that is present in the white region, it is not allowed to form the bond. But there are some exceptions. 2 amino acids we know they are out of this rule. They are glycine and proline. Why? Because glycine has the shortest side chain, that is hydrogenide. So it doesn't qualify for this parameter. And the second one is proline. Proline is also known as helix destabilizing amino acid. So for this 2 amino acids, we are not considering the Ramachandran plot. We will focus only the other amino acids that are present within or without the favorable region. So from the analysis we can see that there are no residues in the white region other than these two. These two are proline. So they are marked in a separate way. Triangle kore mark kora hoyeche.
See the statistics here. So residues in the most favored region, nearly 93% residues are in the most favored regions. So the model, if your model has 90% or more amino acid residues in the most favored region, your model can be considered as a good model. Then there are some residues, nearly 8 or 9 residues in the additionally allowed regions. Those regions are additionally allowed, means those regions are okay, but it is better to have less residues in the allowed region, not in the additionally allowed region. And there is another one that is called generously allowed region. So what are these regions? Allowed region means this is the region if the amino acids are present there are no steric hindrance between the side chains. Additionally allowed region means the steric hindrance cutoff. If you remember how to calculate the steric hindrance, it depends on the distance cutoff, the sizes of the atoms. So if we can extend the distance cutoff a little bit, right? That makes you that gives you the additional allowed regions. Generously allowed regions, if we increase the distance cutoff a little bit more, that is called generously allowed region. Now, these regions, why we are extending these cutoff values? Because if we are very strict, if we are very stringent about these cutoffs, we will never get a good quality model. We will never ever be able to get a good quality model. So, in other words, if the amino acids are present in additional or in generously allowed regions, then also the structure is considered to be a good one. At least you can rely the structure. But if the amino acids are present in the disallowed regions, the structure is not at all reliable. Yes.
On what basis are we considering this generously allowed regions?
The distance cutoff between the amino acid residues.
Up to how much we will?
Generally, the Van der Waals distance is considered. But see, the Van der Waals distance means distance of closest contact. But it is not very strict. Van der Waals distances are not very strict, right? So we can be a little bit flexible.
Means how can we judge that we should be this much flexible and after that we should not go further?
See, if we consider a distance between the center of an atom, the nucleus, and its outermost valence shell, right? That is its atomic radius. Now, the atomic radius of this atom is say like this. Atomic radius of this atom is say like this. Now, these two can come this much close. They are not making any contact. They can come this much close also. They are not making any contact. They can come this much close also. They are not making any contact. So we are considering only that portion. But we cannot consider this. If the distance is like this, it is entirely disallowed. The regions that were made, they were built by considering all types of permutations and combinations between the different atoms of the amino acids.
Sir, what does the significance of the black spots mean?
The black spots means these are the residues which, have a seat, these are the residues which do not actually follow the rules, by like the proline and the glycine. Since proline can take up any, glycine can take up any position, and proline has only some fixed positions because it is a cyclic amino acid. It has got a cyclic side chain, and glycine has only a hydrogen as a side chain. So that is why their positions are not determined by these allowed and disallowed regions. So this is a very important tool, or rather very important process to determine the validity of a structure, right? Or the accuracy of a structure. And remember that you can only plot a Ramachandran diagram if you have the structure. It cannot be obtained from the sequence. You cannot use the amino acid sequence. Okay.
So shall I go for fold recognition and validation method?
Is done. The verification process is done. So, you have any query? This is the homology modeling. This part is the homology modeling. Anything that you want me to
Sir,
clarify?
that means if more glycine or proline analysis is there in such kind of model, then that is not favorable for study.
No, it is not the case. It is not. There is no question of favorable or unfavorable. Glycine and chlorine can take up any positions, but it is not possible to specifically define their locations in the Ramachandran plot. That is why they are not present in the allowed or in the disallowed regions. They are present. They can be present anywhere. So it is not favorable or non-favorable, unfavorable.
Sir, suppose a protein has a polyglycine chain, then what should be this? If we plot it in the Ramachandran pattern, that means we can understand that it is glycine or not. This was the class.
No, that means if you have a polyglycine, it's a polypeptide. Say a polypeptide. In that case, you won't have any amino acid residing in those allowed regions or the gender scale. They will be present somewhere over here. So this is one way of building the model, and the most accurate one in case of bioinformatics. So now the problem arises. Let me go back to my slide again. So see. We performed all these steps. The template recognition and alignment, that is the first step, right? Then the structurally conserved regions, I showed you that 0.93, that portion. Then backward generation loop region and confirmation of start, refinement and the validation. So see, none of these steps you have to perform manually. Everything will be done through a software, a tool. But where the problem then? The problem is, if you give any sequence, you will get any structure. But whether that structure is biologically meaningful or not, that you have to determine. That you have to determine. There lies the challenge. Right? So, let me... Now, I will be talking about the fold recognition, and then we will have kind of a demonstration of fold recognition method. So, fold recognition of threading methods. What do they mean? Fold recognition of threading means, when I was talking about homology modeling, I told you that there is a sequence cutoff, 30%. But it is not always possible to have a template which has 30% sequence identity. Right? Okay. In that case, what will you do? Will you not be able to build a structure? Yes. You will be able to build a structure. And the method of choice is fold recognition of threading. What do they do is that there are no templates available which have 30% sequence identity. But it may so happen that there are certain regions, certain structurally conserved regions, we can build the structures of those structurally conserved regions with the help of this fold recognition or threading method. And then we join those structurally conserved regions with loop. Like this. Say we have a structurally conserved region which contains an alpha helix, then a loop, and then a beta sheet. This is a structurally conserved region. Again, we have another structurally conserved region which has this parallel beta sheets. So we can build the structure of this region, we can build the structure of this region, and then we can join the two with the help of a loop. Right? That means in this case, we do not have a specific template. We do not have a specific template because we do not have 30% sequence identity. But we have certain regions which are conserved in all types of proteins. Right? So we build the structures of those proteins, those regions of the proteins, and then join them together. That is called the fold recognition or threading method. Now, Tanushree will describe the method, and the method that you will get to know, that method is called eye-chaser.
This is the eye-chaser we are doing experiment. So fire 2 looks like this. You have to paste your amino acid sequence. You can provide the job description for your own self, and you can paste as well as upload the file. The file format should contain the extension .pdb. Okay. Without that, it cannot recognize any other documents. Sorry. FASTA or PDB, whatever. FASTA, I'm sorry, not PDB, FASTA. Sorry. It will provide a PDB model. So we will provide the sequence here by pasting it. And you have to be careful when you are working with FASTA sequence, because the little details, single space or a dot or anything, it can ruin the experiment. So for that, you have to be careful to replace these spaces, and the sequence should be like this, a concrete sequence. Okay? So for the sake of simplicity, I am removing all this. It will be easier for the server to identify. Okay? So I am proceeding through by fire search. Nothing internet. Well, it is. The email address that has been provided, the result will be mailed as well as the page will also visualize the result as within a few seconds. So by obtaining the sequence, we are pasting the sequence either as a simple text or we can upload the FASTA sequence, that is dot FASTA, FASTA, and after that we will proceed through file search. It will take some time to go through the repository to find out the exact match for the exact amino acid protein folds present in the sequence. 57 minutes.
See, everyone is using, because this server is being used internationally. So many labs are using this tool simultaneously. So the wait time is nearly 1 hour. So you have to be patient. Or there are options like if you put your email ID, you will get the result in your email. Here, Donshri put her email ID here, so you can put your email ID to get the results in your email. So that is what the preferable method, but that should be done. Otherwise, you have to wait for hours after hours.
So what it will provide, just like Swiss model. Swiss model has provided the templates and the possible model 1, 2, 3, 4 and so on. It will also provide models 1, 2, 3, 4 and so on, but a concrete model by taking all the possible sequences of the old that has been recognized by this algorithm. They will put all the results all together to get the perfect 3D structure of this sequence that has been provided, and this structure can be downloaded as PDF format and we can further analyze it just like we have analyzed through project server. Okay, it will provide the query coverage, the percent identity and the species from where it has been taken the informations, and other attributes will be there to identify which structure or which model we should take, and we will work further with that.
What we learned today is we can generate the structure of an unknown protein from its amino acid sequence. The structure that we generate, we need to validate the structure. And the validation will be done with the help of the tool project, where we plot the Ramachandran diagram. And in the Ramachandran diagram, from the Ramachandran diagram, we could gather which amino acids or how many amino acids are present in the allowed region, how many amino acids are present in the disallowed region. And as I told you that no amino acid should be present in the disallowed region to build a good structure, to have a good structure. Right? So it is also the same case here. In prior to this, the method of building the structure is different. The previously we used the technique of homology modeling or comparative modeling, since we have 30% sequence identity. But here, whenever we do not have 30% sequence identity, we go for this fold recognition method, that is called Bioinformatics. So here the structure will look the same as before. You can perform the same project analysis, all those analysis to get your structure. But it is always preferable to use your email ID instead of this interactive page. Okay, because sometimes it takes 1 or 2 days to complete the entire process. It depends on the load on the server. So let the result come. Mediation.
Okay. It contains the confidence score 100%. Query coverage is 99%, which is a good thing. The template name is provided here as well, and the amino acid residue, total amino acid residue for this chain was 147, and in both the 145 residues have been modeled with 100% confidence score. So to obtain this model, we will just click on this and তাহলে to download the model, we will left click this one and the model has been downloaded as finalcast.pdb. Okay, so একবার খুলে দেখে নিই এর মধ্যে কি কি ইনফরমেশনস আছে. Yes, it contains the atomic coordinates of the amino acids. The XYZ coordinates are present here, and the amino acid sequence is perfectly mentioned. And the important thing is the start and end, which is not here present. You can add it to get a concrete model. So, after obtaining this model, we will again check for Ramachandran plot analysis using saves. For that, we are again choosing the .pdb extension that has been obtained from file to final.casp. Open, run programs. Pro-check.
Have any doubt, you can clear it up, and then you will have your lunch outside. And you can suggest for any topic that you would like to learn, and so we will consider those topics in the next workshops that we will conduct. So it's from these workshops are for the students. So any topic that you want us to explain to you, we will conduct the next couple of workshops on those topics. Okay, and one thing is we have our social media handles. We have our Facebook and Instagram and Twitter, that is X. So you can follow us, you can subscribe, and you can log into those accounts, and you will get to know about the upcoming workshops and the different tutorials that we post over there, different videos and tutorials that we keep on posting in those social media platforms. So you'll be able to find it out. This is the Bioinformatics Infrastructure Facility Center of Kolkata University.
So the results are here. I will go for the Ramanujan plot analysis, which shows a very good model. How? Because it contains the residues, maximum residues are in the most favored region, containing 93.5% of number of residues. That is 145 residues are there. These are the residues. Most of the residues are in the red region and the dark yellow region, or this one, and one proline residue is outside, which is in the most disallowed region, but it will not be counted. As we have said, proline and glycine, they are exempted from the study of Ramanujan plot analysis. So the most important thing is how many residues are there in the disallowed region? 0. So it is a good model. And how many residues are there in the most favored region? 93.5. That is more than 90%. So this is the best model, and it is this server provided a model without any amino acid residue in disallowed region. Okay. So as a conclusion, it can be said that homology modeling is based on the homology or the structural alignment prediction, and then further it provides a model which contains particular homology or sequence similarity with other proteins belonging from the same species or different species. In case of fold recognition, the sequence identity is less than 30%, so particular homology for that particular sequence is not found, so we go for fold recognition method, and this is also useful to identify unknown protein from other species or variations. So for identifying an unknown protein and its possible function, the fold recognition method is advised, and for a known protein but its structural other features to be understood, we should go for further homology model.
So, with these few things, I'm going to conclude the session. And your lunch is outside. You collect your lunch packets and then provide your feedbacks. Your feedbacks are really valuable for all of us. And any topic, you can tell me any topic that you want us to teach you, so that we will conduct the next workshops on those topics. Anything that you require. Okay, that means docking and all this. That means docking and all this. Okay, so we'll consider that in the next workshops. So thank you all. Thank you for coming here, and I wish you all the best.